Welcome you all. I'm Dr. Nitya, Assistant Professor in Department of Botany, Sita Lakshmi Ramasamy College, Tiruchirappalli. Now I'm going to explain about protoplast isolation and culture. Protoplast is nothing that is a plant cell without their cell wall surrounded by only plasma membrane. The plant cell is the basic structural and functional unit of plant. Like all eukaryotic cells, it contains nucleus and various organelles enclosed within a membrane. So this is the right side. You can see the protoplast, and the left side you can see the normal plant cell with the cell wall. Now let's see what is protoplast. Protoplast is a living plant cells from which the cell wall has been removed either enzymatically or mechanically. The protoplasts are spherical in nature due to their absence of rigid cell wall. Why isolate protoplast? The protoplast which helps to facilitate genetic manipulation, cell fusion and study of cellular processes without the interference of the cell wall. The sources of protoplast. We can take the protoplast from leaf mesophyll cells, callus tissues, suspension culture and root tips. Methods of protoplast isolation. There are two different methods of protoplast isolations are there. One is mechanical method, another one is enzymatic method. In enzymatic method, there are two subdivisions are there. One is sequential method, another one is simultaneous method. Mechanical isolation method. The mechanical method is an older and less commonly used technique to isolate the protoplast. It does not involve any enzyme, but instead of that, it relies on physical disruption to remove the cell wall. It is widely used for large celled algae. Limitations of the mechanical method. Mechanical method which gives very low yield of viable protoplast, and it is also labor intensive and time consuming. It may often damages the protoplast. It is only suitable for certain large celled algae, and it is not widely used for higher plants. Now let's see the enzymatic isolation method. The enzymatic isolation method is most widely used method, and it is effective technique for isolating the protoplast from plant tissue without any burden. It involves using the cell wall degrading enzyme to gently remove the rigid cell wall while keeping the cell membrane and contents intact. First, let's see the sequential method. The sequential method, it is actually done by the first we can remove the middle lamella, and then the primary cell wall is removed with the help of some digestive enzyme. Second one is a simultaneous method. Here, the both middle lamella and primary cell wall are removed in a single step using a mixture of enzyme. Steps involved in enzymatic isolation. The first one is tissue selection, next plasmolysis, enzymatic digestion, filtration and centrifugation, washing and resuspension. Final one is viability checking. Selection of plant tissue. Young and soft tissue can be used to isolate the protoplast because it is ideal due to thin wall. The next step is the plasmolysis. It is actually optional. The tissues are placed in a hypertonic solution like mannitol or sorbitol, which is used to cause the plasma membrane to shrink away from the cell wall. This reduces the cell damages during the digestion. Next step is the enzymatic digestion. The tissues are incubated in a solution containing pectinase and cellulase. The pectinase, which is otherwise called as macerozyme, this is an enzyme which is used to digest the pectin in the middle lamella. Cellulase is an enzyme which break down the cellulose, and we have to keep it in 25 to 30 degree Celsius for 1 to 4 hours. The next step is the filtration through mesh and centrifugation using sucrose or mannitol gradient to separate viable protoplast. The digested material is filtered through the fine mesh, example nylon mesh, to remove the debris and undigested tissue. The next step is the centrifugation. The filtrate is centrifuged to pellet the protoplast, often done using the density gradient centrifuge. Example, sucrose and mannitol which is used to separate the viable protoplast. The next step, the protoplast can be washed and resuspended in an osmotic stabilizing solution to prevent the bursting. Viability checking is the important step which is involved in the enzymatic isolation. The protoplasts are checked their viability with the following common method. One is fluorescent diacetate staining, propydium iodide staining, and trypan blue exclusion. Fluorescent diacetate staining. It is a method which detects the living cells by it emits a green fluorescence light. The living protoplasts have active esterase that converts non-fluorescent FDA into fluorescent fluorescein, which accumulates in the cells with the intact membrane. So the results wherever you can see the living cells which emits green color, and if there is no fluorescence which remains, that's the dead cells. Next one is the propydium iodide staining, which is used to detect the dead cells by red fluorescence, because the propydium iodide cannot cross intact the cell membrane. So only membrane compromised, that is dead cells only, which is take the stain. So the result shows that the red color cells which consider as a dead cells, and the living cells does not take the stain, so that it is unstained in nature. Next one is trypan blue exclusion. It is simple bright field method without the fluorescence. The principle, living cells exclude the dye, the dead cells take it up. So the results, that is living cells which is unstained in nature, and the blue cells which shows that the dead cells. Advantages of enzymatic method: it is actually gentle and very effective, and which gives high yield of viable protoplasts and suitable for wide variety of plant tissues. Applications of protoplast isolation. The genetic engineering, that is protoplasts allow direct DNA transfer into plant cells, making them ideal for producing genetically modified plants. Somatic hybridization. Fusion of protoplasts from different species can be create novel hybrids. For example, pomato. This is a pomato, and which is a fusion of potato and tomato, which is not possible through the traditional breeding. So it can be done only through the somatic hybridization. Study of cellular process. Without the rigid cell wall, the scientist can easily study the membrane biology, ion transport, and signal transduction. And the single cell studies like protoplasts enable the analysis of cell metabolism, gene expression, and enzyme activity at the single cell level. Plant regeneration. The protoplast can regenerate into whole plant through the tissue culture, useful in cloning and micropropagation. And it is also used for the drug and pathogen testing. They provide control system to test the effect of chemical, drugs or pathogen directly on plant cells. Challenges in protoplast culture. The regeneration difficulties. the cost of the enzyme, and genetic instability and contamination risk. These are the challenges which is caused in the protoplast culture. Now the conclusion. The protoplast isolation is a key tool in plant biotechnology, enabling advanced genetic, cellular and developmental studies. Thank you for watching this video. Thank you once again.